Hi everyone, welcome to Unit 2, Chapter 2. We're going to learn more about the cell membrane. Last week we touched on the cell membrane, and the week before last we talked about phospholipids and how they create a double bilayer. This week we're going to go into detail about what that means for your cells. As always, if you need the guided notes, you can click on this icon right here. This week, we're going to be discussing the fluid mosaic model of the plasma membrane. We're going to talk about different structures within the plasma membrane and their functions. And then we're going to talk about different ways that things can cross the plasma membrane, so different types of transport. The plasma membrane functions to define the outer border of all cells and most organelles. Last week we learned about membrane-bound organelles such as the endoplasmic reticulum or the Golgi apparatus. Those are made of a phospholipid bilayer. In fact, vesicles are pinched off pieces of the lumen or the phospholipid bilayer of those organelles. They manage what enters and exits. So the phospholipid bilayer is mostly impermeable unless certain conditions are met. And it also helps to receive and send signals to other neighboring cells. Phospholipid is very unique because it's amphipathic, meaning it has a hydrophilic end with this phosphate group and a hydrophobic end with these fatty acid tails. So it automatically orients to the center, creating a double bilayer. The plasma membrane is what's called selectively permeable, meaning it gets to choose what enters and exits the cell, which is really, really great. Some small nonpolar molecules can pass really easily because they're not rejected by these tails, okay? So small dissolved gases, for instance, they're nonpolar, they can cross. Large things and anything that's polar, like water, is going to have to cross through some sort of carrier protein or channel protein. The phospholipids and proteins are always oriented in this way where the hydrophobic tails are pointing inwards and the hydrophilic heads are pointing outwards. Because cells are always in an aqueous solution. There's water on the outside, there's water on the inside. So it automatically orients in this double bilayer. The fluid mosaic model was proposed in 1972 by S.J. Singer and G.L. Nicholson, and they basically just said that the phospholipid bilayer isn't the only thing that's going on here. We also have lots of other cool structures that are embedded in the bilayer. So let's watch this video. So what else we have here is in the fluid mosaic model, we're not saying that it's just made out of phospholipids. There's also cholesterol. And remember when we talked about cholesterol, it is a lipid, but it helps to act as a heat protectant, preventing the phospholipids from becoming too fluid or too viscous. We also have integral proteins. Integral proteins are proteins that span the distance of the phospholipid bilayer. We also have peripheral proteins, which are only on one side of the phospholipid bilayer. They could be on the inside or the outside. And we'll talk more about integral proteins and peripheral proteins and the jobs that they do in the next slide. We also have carbohydrates on the outside surface. We don't see carbohydrates on the inside surface, mainly just on the outside surface in the form of glycolipids that are connected to a phospholipid, or glycoproteins, which are connected to a protein. Proteins are the second most abundant part of the phospholipid bilayer of the plasma membrane. Lots and lots of proteins, and they come in two main categories. Integral proteins are integrated completely into the phospholipid bilayer. They're able to do this because they are hydrophobic, or a region of them is hydrophobic, which causes them to sit in between the phospholipids because they are trying to get away from the water on either side. Okay. And the outsides are going to be made of amino acids, which are hydrophilic. So the outside of it can touch the water, but the inside does not want to touch anything aqueous, so it sits in between the phospholipids. They can do different jobs. For instance, this looks like a channel protein or a carrier protein. This one looks like maybe some kind of signal receptor. They do different jobs within the cell membrane. And remember, the fluid mosaic model states that they can kind of move around a little bit. Then we have something called a peripheral protein, which means it only occurs on one or the other surface. It won't cross the entire phospholipid bilayer. It will just be sitting on top. The carbohydrates are also important to the phospholipid bilayer. Carbohydrates are located on the exterior surface. They're not going to be found on the inside of the cell. They're going to be found mostly on the outside, and they're important for cell signaling or cell-to-cell recognition. So your cells can recognize other cells that are yours and not attack them because they have these glycolipids and glycoproteins. So glycoproteins are made up of a carbohydrate connected to a protein, hence glycoprotein, and glycolipids are made up of a carbohydrate connected to a lipid, so hence the name. One very important glycolipid is going to be your blood type, glycoporins, okay? They send a signal that the blood is going to be a certain type of blood. So for instance, you're going to create antigen O, or if you're blood type A, you'll make antigen A and antigen O. And if you're type B, you'll make antigen B and antigen O. If you're type AB, you will make all three of them. And that means that you won't produce antibodies against that type of antigen. What this results in is blood typing. So for instance, blood type A can donate to A or AB because you don't produce antibodies against A. But if you produce antibodies against it, for instance, blood type B would produce antibodies against A, so B blood can only be donated to B recipients or AB recipients. Type O is going to be the universal donor. Anyone can receive type O. You'll see here it goes to O, A, B, and AB. And type AB is going to be the universal recipient, meaning that they can receive type B blood, type O blood, or type A blood because of this surface signal. See, AB can only be donated to AB, but AB could receive anything. AB can receive anything. The structure is going to result in it being selectively permeable, meaning it only allows some things through the membrane and not others. Non-polar substances that are hydrophobic, can cross the membrane more easily, especially if they're very small, such as dissolved gases, because they are able to be around these tails, these fatty acid tails. Larger substances are going to need help crossing, especially if they're polar, including water. Water does not cross here. Water has to cross through something called an aquaporin. An aquaporin is a complex of several different proteins which creates a pore that water can cross through. So membranes are generally considered permeable to water only because aquaporins, not because they can cross right here. This means that cytosol inside the cell can have a different composition to the extracellular fluid. How do things cross? Well, selectively permeable membranes have several different methods of getting things across the membrane. They may move through the plasma membrane through passive processes, meaning that it does not require any energy input. Does not require energy. It's not going to take ATP. It's not going to take any sort of energy to do it. It's only going to go down a concentration gradient. Active transport processes require energy. It's going to require energy usually in the form of ATP, but not always. We'll talk about why. Passive transport includes diffusion. Diffusion is just direct molecules being able to cross the membrane. So this is only going to be very small, non-polar molecules like dissolved gases or maybe some lipid hormones because they're able to cross these fatty acid tails. Now, diffusion always goes down the concentration gradient. That means that if we have a whole bunch of substance on one side, it will kind of spread out and go to the lower side until it reaches equilibrium. So here in this image, we had a whole bunch of this molecule on this side. It is able to cross the phospholipid bilayer directly, which leads me to believe that it's either oxygen, carbon dioxide, or some kind of fat-based hormone. And it's going to move to this open area because it's not as crowded over here. But once both sides become equally crowded, or we have reached equilibrium, then it will cross with no net movement, meaning this one can cross this way and this one can cross this way, kind of at equal rates. Factors that affect diffusion rates are factors that you think of pretty frequently. Concentration gradients for one. If we have a really different concentration gradient, then the diffusion will happen faster. The mass of the molecules is important. Smaller molecules are going to diffuse across that membrane a lot faster because it's a lot easier for them to cross. We also have temperature. I always use the example, if I'm making tea and I'm trying to dissolve the sugar, am I going to use hot water or cold water? Well, warmer molecules have higher energy and move faster. Surface to volume ratio. Surface to volume ratio is really, really important. And this is another thing, this is another reason cells are small, is because there is an upper limit of cell size based on surface to volume ratio. Pressure is also really important, and solubility. So is it soluble in water? We can look at the facilitated passive transport. So that was just diffusion, but facilitated transport still does not require any energy, but it does require a protein to help cross the barrier. So this is going to be slightly larger molecules or maybe polar molecules that can't cross directly across the membrane, such as ions. So carrier proteins are going to bind to that substance and change shape to be able to carry it. It may allow movement in either direction, but it still uses the concentration gradient. So we're still going from an area of high concentration to an area of low concentration, but it's going to kind of manipulate its shape. So the protein is actually changing shape. Whereas a channel protein is going to just be open, and sometimes they're open all the time, like aquaporins. Aquaporins are always open, and water can move freely from one side to the other depending on the concentration gradient. But others are gated, meaning they have to have some sort of signal. For instance, action potentials or muscle contractions are gated channels, and so they only open when there's a certain charge present or a certain molecule present. And this is facilitated passive transport, still not using any energy. Osmosis is another type of passive transport. Osmosis only deals with water. We're only focusing on water. So we're going to be talking about aquaporins. Water is always going to move from an area of greater water concentration to an area of lower water concentration. But that makes it seem like we're moving from an area of less concentration of solute to an area of higher concentration of solute. But what it's doing is it's trying to spread out all of that solute. So the water moves this direction to the more, to the higher concentration of sugar side, and you end up with an even concentration of sugar on either side due to the osmotic pressure, pushing water here to kind of dilute all of those sugars. Osmoregulation is the active regulation of osmotic pressure in an organism's body in order to achieve homeostasis. So in order for your cells to survive, they have to manage how much water enters and exits. Osmosis is still going to go from an area of high concentration to low concentration, but keep in mind that we're talking about high concentration of water molecules, not solutes. Tonicity is describing osmosis. So it's describing the extracellular fluid in relationship to the intracellular fluid. There's 3 different types of tonicity. Hypertonic, hyper meaning higher, has lots and lots and lots of solutes. So we're going to have, for instance, salt water. The water will actually leave the cell because the water outside of it is so salty. Okay, so there's less solutes inside the cell and there's more solutes outside the cell. So the water tries to leave the cell to adjust for that. That can be really bad for animal cells and plant cells because it can cause them to shrink and shrivel or crenate. In plants, this causes it to plasmolyze, which means that the cell membrane is separating from the cell wall. Isotonic, iso meaning the same, means that the extracellular fluid has the same amount of solutes as the intracellular fluid, and there's no net movement of water. Water moves into the cell and out of the cell at the same rate. This is perfect for animal cells. Animal cells want to be isotonic. Plant cells, however, don't really want to be isotonic. This is going to cause the plant to become flaccid or wilted. The last one is hypotonic. Hypotonic, this is supposed to be an O. Hypotonic means that the extracellular fluid has more water and the inside of the cell has more solutes. So the water is going to try to go into the cell, and this is really bad for animal cells because it can cause them to burst or lyse. This is great for plants because they become turgid or stiff. Okay, so hypertonic means that the cells are going to shrink because the water leaves the cell. Isotonic means that the water is going to leave and enter the cell at the same rate, and hypotonic means that the water is going to enter into the cell. So in this example, this cell is in a saltwater solution similar to ocean water. Is this a hypertonic, isotonic, or hypotonic cell? Well, if this egg represents our cell and we have salt, the salt cannot cross this membrane, but the water can. So the water from inside of the cell is going to move out to try to dilute all that salt. So this is actually a hypertonic solution, and the cell, or the egg in this case, will shrivel. This cell is in a solution called Ringer's solution, which is an isotonic solution because we have the same amount of solutes inside the cell as outside the cell. So there will be equal number of water going in and out at the same time, so no net movement. This cell is inside distilled water. It has absolutely no solutes, but inside of this egg is protein and cholesterol and all kinds of stuff. So the water actually will try to force its way into the egg, and the egg will expand or become lysed. It will kind of swell. So it is a hypotonic solution. That's why we shouldn't ever drink distilled water, because we don't want this to happen to our blood cells. So, which plant here has lost turgor? The wilted plant has lost turgor. Once you water the plant, it will return back to its turgid state. But this one right here has lost turgor because it has lost water. Osmoregulation happens in several different ways depending on what type of organism you're talking about. Terrestrial organisms are going to, conserve and hold their water inside of their bodies. But aqueous, like fish, marine animals are going to have different methods of regulation. So, for instance, this freshwater fish is going to urinate very dilute urine, whereas the marine fish is going to urinate very concentrated urine that has lots of salt in it. Protists, like the paramecia or amoeba, is going to use vacuoles to pump water out of their cells so that they don't burst. So water is constantly being pushed into the cell, and it has these contractile vacuoles that act as a pump to pump it out. Active transport requires energy, and that energy can come in two different ways. Active transport is going to move things away from the concentration gradient or away from the electrochemical gradient. This image here represents an electrochemical gradient where we have an area of very positive charge and an area of very negative charge. So even though there's more potassium on this side, since it's positive, the potassium, if it were allowed to cross, would force this direction because it's trying to get away from the positive charge. It'll move this way, even though we have a lot of that's pushing too much potassium there. It's going to work that direction because of the positive charge here. Or we can go against the concentration gradient where we're going from an area of low concentration to an area of high concentration. And in this case, that takes energy in the form of ATP. This is called primary active transport because we use ATP. Secondary active transport is going to use an electrochemical gradient kind of like this to do a job. But it's called secondary active transport because they had to do this first, and we'll go over an example of that in a second. Primary active transport, such as the sodium-potassium pump, is going to use ATP. The sodium-potassium pump takes 3 sodium and 1 phosphate and pumps that out of the cell. And meanwhile, 3 sodium are going to exit the cell and 2 potassium are going to come in. And every single time this sodium-potassium pump changes shape, it has to use energy from an ATP. So you'll see this last phosphate breaking off, releasing energy, which changes the shape and allows 2 potassium to come in while 3 sodium exit. So 3 sodium exit while 2 potassium come in. This is an example of primary active transport because it directly uses ATP. Now the reason why we want to do this is because now we've created a chemical gradient where we have tons and tons of positive sodium on the outside and we have positive potassium on the inside, but it's not nearly as much because we're pumping more sodium out than we're pumping potassium in. And we allow some of that potassium to leak back out, which means that we have even more positive on the outside and more negative on the inside. This sets us up perfectly for secondary active transport. So since we have so many sodium on the outside creating a very positive charge, now if we were to open that barrier, all of those positive ions would be rushing back inside. And this is how action potentials work. ATP synthase also works this way. The electron transport chain does a great job of pumping hydrogen into the intermembrane space, and then that hydrogen flows back into the mitochondrial matrix through this ATPase complex, which adds a phosphate to an ADP every single time a proton or a hydrogen runs through the system. So we created the electrochemical gradient and pumped all of these protons up here just so that they could flow back through and power this engine to create more ATP. That is secondary active transport. Transport comes in different forms. You can have a uniporter, which is going to carry a molecule one direction. A symporter carries two different molecules in the same direction. And an antiporter carries two different molecules in opposite directions. So the sodium-potassium pump would be an example of an antiporter because we have sodium, 3 sodium leaving, and 2 potassium entering at the same time. So it says, in which step does hydrolysis of ATP occur? So ATP is going to join the ATP complex, or this sodium-potassium pump complex. 3 sodium are really attracted to it, so they join in there. Once that ATP joins, it becomes hydrolyzed, or hydrolysis happens, breaking off one of those phosphates, releasing energy and changing the shape of the protein. Now sodium is no longer attracted in there, it will get rejected, but potassium is now attracted to it. So potassium will join, that phosphate kind of breaks off because it's no longer being used, and we go back to our original orientation, which will reject the potassium but attract the sodium, starting the process all over again. So this is the point where ATP becomes hydrolyzed. An electrochemical gradient like the one shown is used in what kind of transport? Well, it's going to be used in secondary active transport. Which transport protein is a symporter? Well, we already talked about an antiporter, the sodium-potassium pump, and a uniporter is only going to push one thing through in one direction. So this would be the uniporter. The symporter is right here. It's going to pull these protons or pull these hydrogens in at the same time that they pull in sucrose. So this would be the symporter. Another type of active transport is going to be bulk transport. It does require energy, but I don't require you to memorize the mechanism in which it uses energy. But basically, the cytoskeleton will help to stretch and move the phospholipid bilayer to consume. They create these little pseudopods, these little feet right here, to consume or bring in nutrients. So endocytosis means inside. It's bringing things inside, such as phagocytosis, where we're taking and engulfing extracellular substances like a phagosome. Okay, so we're going to create a little phagosome, a little food particle is being pulled into this phagosome. It just pinches off the phospholipid bilayer and creates its own little vesicle. Another type of endocytosis is called pinocytosis. It's where a divot is created and it kind of creates a suction and sucks liquids in. So this is pinocytosis is mainly for liquids and dissolved nutrients, where phagocytosis is larger substances. Phagocytosis points outwards, whereas pinocytosis points inwards. Exocytosis is exiting. It's the opposite of endocytosis. It's when we're pushing items out of the cell. So here is a picture of phagocytosis, and we can watch a short video of phagocytosis. Here we can see pinocytosis. Pinocytosis starts out with this divot right here, and as that divot increases in size, So as that divot gets larger, it creates kind of a suction, and then we pull it in, creating its own transport vesicle. Here we have receptor-mediated endocytosis. All right, so receptor-mediated endocytosis is most similar to phagocytosis, except that we have to have a signal from the receptor in order to create that vacuole. In this exercise, I was asking my students to identify different types of bulk transport. Remember, bulk transport is still active transport. So receptor-mediated, of course, is going to be this one. Phagocytosis is going to be this one. Pinocytosis is going to be this one. And then exocytosis is exiting. It's going to be where the vesicle exits by fusing with the membrane and then pushing its contents outward. Thank you for joining me this week. As always, don't forget you can click on this icon and get a practice homework, an optional practice homework where you can watch videos and do a drag and drop activity. Don't forget that you do need to click on this and print it out. You can either complete it at home by scanning the QR code and completing the activity, or bring the activity sheet to class if you have face-to-face lab. Don't forget that you have a chapter quiz due Friday at 5 p.m. And as always, we have all of these extra resources if you want to use the textbook, the flashcards, or the games. Also, the practice quizzes is available outside of this PowerPoint.